the glycoproteins and the lipids on the outside, which can attach to the extracellular matrix, but they also can tag it as a specific type of a cell, so we can recognize cells. Yeah, Colleen.
So is that all enzymes do? Is just break things down?
They do, and they build things up. Yeah. So what is a ribosome? Ribosome is like a big enzyme. That's basically what it is. It's an enzyme that can have multiple different shapes as the RNA goes through it. Yeah. So they're made to build and they're made to break. Yeah. If we look at this, what am I kind of modeling right here?
Osmosis.
Osmosis, right? Diffusion's okay, but we're looking at diffusion of water over time. Are we good on this slide and what it means? All right, and so when are animal cells happy? When we are isotonic with our environment. When are we sad? When we're hypo or hypertonic. So if I want to kill somebody, I can inject distilled water into their veins, and all of their red blood cells would burst. Yeah, I won't do that. When you go to the doctor, though, they don't give you distilled water, they give you saline solution, so they're going to make sure that it's the same exact concentration across. When are you happy with your plant? When you're inflated, but they have a cell wall.
How much distilled water do you have to inject into someone?
Yeah, we'll talk about it later when we're planning to do some people later.
Yeah,
Joe.
creative ways. So I was wondering, when it's hypotonic, is that the environment hypotonic or the cell?
That's a good question. So it's relative to the cell. And so it's saying here that it's in a hypotonic solution. Yeah.
All right, I get it now.
And we could say that this is hypertonic to the solution, so it's relative.
All right.
Why is there a difference between here and here? Why do we die and they don't? Cell wall, right? Can you see how this is what we did in the potato lab? Put them in distilled water, they got big. Put them in sugar water, and they got small. Yeah. Are we good on all these types of movements? Let's go perfect on this. See if we can run it. All right? As a class, what is this movement? Diffusion. Okay, we're perfect so far. What about this one? Oh, somebody said active. Who was that? Facilitated diffusion. How do we know it's facilitated diffusion and not active transport? Right, but we could do better, right? The gradient is in this direction. Can you see that? There's more here than there. If it was moving stuff in this direction, then we call it active transport. Yeah. This is sodium potassium pump. Is it active or passive?
Active.
Active, right? Yeah, it's hard to move ions. And then, what are these things? What are all of these three things? Endocytosis. We got food, we got water, or we got specific molecules. And exocytosis is where we let it go. And then 17 is basically about compartmentalization. So cells increase surface area so they can increase the amount of chemical reactions around it, and diffusion doesn't have to take as long. But if we were to talk about parts of the cell, where are we going to make proteins, for example? Where would that be? Just yell it out. Where are we going to make the proteins? Rough ER, but we also are going to have free ribosomes in here as well. Where does the energy come from? Mitochondria, right? Glycolysis, Krebs cycle, electron transport chain. Yeah. What dissolved material in the cell? Lysosomes have enzymes inside it, so it can break material down. But also we could break it down in smooth ER as well. Yeah. If we had a prokaryotic cell, would it be bigger or smaller? Quite small, right? About the size of the mitochondria. Yeah. What is that going to have? What is a prokaryotic cell going to have on the inside of it? Nucleus? Yay or nay? No. Yeah. Is it going to have DNA though? In a nucleoid region. Is it going to have cytoplasm? Is it going to have ribosomes? Yes. Is it going to have a cell membrane? Yes. Is it going to have Golgi apparatus? No, right? Yeah. So we don't have compartmentalization. And so basically what we get, and again, somebody thought this up yesterday, I don't claim credit. Compartmentalization of a cell is like compartmentalization of a school. Biology will always be taught here. English will always be taught here. Spanish will always be taught here. The main office will always be here. Is that somebody in here come up with that? Was that you? Thank you. That was good. I'm loving it. I'm beating it to death. Okay? So compartmentalization is having all these different things in different places. So if we extend your analogy then, a prokaryotic cell then would be more like a...
Hot tub schoolhouse.
Not like a hot tub. It still has to do all this, biology, math. I think it's like a one-room schoolhouse. You know what I'm saying? Yeah.
It's not a hot tub.
Yeah. It's funny. Yeah. So how does it make energy, though? I mean, this is a question that everybody misses until I walk them through it. Like, how does it make energy if it doesn't have a mitochondria? It uses the cell membrane, right? Glycolysis, Krebs cycle, electron transport chain. It breaks down the sugars outside the cell in the environment. Questions on everything we talked about?
Like,
What's that?
how is it breaking down the stuff if it doesn't have like enzymes outside of the
It
cell?
does have enzymes outside the cell.
Oh, it does?
Yeah, it secretes enzymes outside the
Okay.
cell. How does the glucose get there? I mean, it is a really deep question. Like, how's the glucose getting there? Diffusion, right? Because we're gobbling up all the glucose there, so it's just moving there. Yeah. And it probably makes it it's even more effective because we don't have to bring all of that necessarily across that first membrane. Okay, so if you want to review, I'll do a review of 17 at first, maybe. If you need to do microscopes or anything else, let's do that. Big questions or not?
The test was due last night, right?
If you have to finish stuff today, that's okay. Yeah. There's so many of you who have like 20 points for everything, but like 6 points for 17. Finish it out. Make sure you get all your points so you don't lose anything for the big test. If you want to do 17 review,